Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, the topic is related to microbiology, and the title of my today's lecture is that how will you identify a microorganism in the lab? In this video lecture, I will discuss all the steps that how you will identify a microorganism in a specified sample. For example, if your examiner give you a sample and ask you that identify the microorganism that are present in this sample. So how will you start the procedure and how you will end it? Okay. So in this video, I will discuss all the steps in a brief way, so stay, watch the video till the end, and at the last, I'm pretty sure that it will help you a lot in your assignments and your exams. Okay, so let's start with the very beginning. For example, you are working in a laboratory, and you are asked to collect a sample from a patient. Okay, there is a patient. So in this slide, we have discussed that I will discuss that how you will take the sample from a patient. For example, if he is willing to give, if you are testing his sputum, so you have to collect the sputum from his nasal cavity or from his mouth through a sterile swab and place it in a tube. Or if you are testing his blood, so you have to suck the blood through syringe, and after that you have to pour the blood in a vial, and then you have to test it. If it is of urine test, so then you have to give a bottle to your patient, and he have to place his urine in that bottle. And after that, what will you do? You will place the sample in a sealed plastic bag with a paperwork that which type of test you are conducting, and other the patient name and different other type of stuffs. Okay, so this is the second step. After that, now this is your work. What will you do when you carry the sample into the laboratory? So the first thing is that you have to increase the population of that microorganism so that you can easily detect it, okay, through microscope, because in the sample it could not be directly detected. So what will you do? You have to first prepare a nutrient media or a culture media for that microorganism. So this is a very brief procedure. I think most of you will know about it, that how you will prepare a culture media. You have to take a culture media and you have to add distilled water to it, and after that you have to dissolve that in a flask, and then you have to keep that flask in an autoclave just to eradicate all the microorganism that is present as a contaminants. Okay, after that, then you have to take plastic petri plates. They are already in a sealed plastic, and then you have to pour the media into that into that petri plates. And I'm pretty sure that this procedure you will be knowing about it, because this is very the starting of the microbiology student, that he should know about this procedure. After that, after autoclaving, you have to take an inoculum, and you have to streak that inoculum on the petri plate, just like the streaking that is here in the this diagram, the first step. And after that, this is an just look to my cursor, this is an inoculating loop, and then you have to streak it on a on that petri plate. And how you will do how you will perform streaking? So I'm pretty sure that you will know about it, that how you will perform streaking. Just like this, first you have to streak that microorganism in different direction. It's not necessary that you have to keep a specific direction, but just keep this thing in mind, that in which direction you have streaked the microorganism. After streaking, you have to place the petri plate in an incubator. Why you are placing it in incubator? Just to increase the population of that microorganism. And incubator provide the perfect environment for the growth of the microorganism. After that, when there is a growth in the of the microorganism, so the first step is that you have to do for the identification is the gram staining. And this is the procedure of the gram staining. You have to take a small sample and place it over a glass slide, and then you have to add different dyes to it. and process the complete procedure with mechanism of gram staining. I have uploaded a video over it, so search that video in my channel, and you will find it that what is the actual what are the actual steps of the gram staining. So through gram staining, you will came to know that either my microorganism is gram positive or gram negative. If it appears pink, so you will then you will conclude that my microorganism is gram negative, or if it is purple in color, so then it will be gram positive. So from this test, you will identify that either it is gram positive or gram negative. After that, one thing I want to tell you guys that if you are an experienced microbiologist, so from the growth of the from the growth of the microorganism, you can also detect that which type of microorganism I am dealing with. Up to 40 to 50% you will came to know. Just like this is the colony formation, that what is its form. You will check the form and then you will compare it with a scale. Okay, these types of charts are placed in the laboratory, so then you can compare your microorganism, your actual growth of the microorganism with this chart. Either it is circular, the form is circular, filamentous, irregular, rhizoid, spindle, or how is the elevation, what are the margins. Okay, so these are such type of colony formation. So different microorganism forms different colony. So from this you can detect that how what is the morphology of the colony of the microorganism. So from here you can also detect that I am dealing with this such type of microorganism, but it is not that much accurate. Okay, after that, what will you do? If you look here, so you can also detect the motility of the microorganism with the help of microscope. What will you do? You will take an inoculum, and after that you will streak it over a glass slide and add a few drops of water into the glass slide, and then check it on the on the microscope. If your cells are moving, so you can say that they are motile, or if they contain flagella, so you can easily say that my cells are motile. So it can also give an information about the microorganism that I am dealing with a motile microorganism. You can also detect the structure of the microorganism, that either through microscope, that either it is round shape, or it is bacillus in shape, rod shaped, or it is spiral in shape. So through microscope you can detect the mobility of the microorganism, the actual morphology of the microorganism, and by the naked eye, looking to the slide, looking to the petri plate, you can detect the how the formation of the colony, that how it has formed the colony. So from these information you can easily tell that it can give you a very sufficient information about the microorganism, but it is not enough for this. After that you have to take the special colony from it, that you have detected that that this should be my colony. And after that you have to perform different biochemical tests, and these are the very actual procedure to identify the microorganism. Biochemical tests are very much easy to perform. Most of the tests that we perform is actually take 5 to 10 minutes, and in case of antibiotic sensitivity or fluorescent pigment, it takes a little bit time, a day or two. Okay, so and this is a special chart, and it must be present in all of the microbiology lab. You have to compare your result with this chart. Okay, for example, you are dealing with pseudomonas, so you have checked the gram positive and the gram negative. Okay, you have done the gram staining, you have checked the cell shape, the spores, and the motility through microscope. And after that, after from here then you will perform different tests. So you can check the antibiotic sensitivity, that which antibiotic is sensitive to to which antibiotic microorganism are showing sensitivity. Okay, so this is also a very good information. And after that you have to perform oxidase test. It is very much easy, and it can be done within 10 minutes. Okay, oxidase test, catalase test, I will make videos over these tests. Okay, but just keep this overview and this background in your mind. So Oxidase test, catalase test also take about 10 to 15 minutes. Opacity factor test, okay, this is also a very small type of test. Urease test, gelatinase test, and fluorescent pigment test. So through these tests, by performing all these tests, you will able to know that I am dealing with the species of the Pseudomonas. Okay, for example, all your results are, when you compare all your result with this standard, so you, and if it is 100% correct, so you can easily say that I am dealing with the Pseudomonas species. Up till here, working in a microbiology lab, it is enough to identify a microorganism. Okay, or if you want to proceed further and you want to detect the actual species of the microorganism, so then you will proceed further. But when you are working in a microbiology lab, so this is enough for you. Okay, up till 90% you can easily clearly tell that I am dealing with the species of the Pseudomonas. Okay, but if you want to check the actual, then after that you have to send the results with the help of a computer, and after that you will give you will give the results to your patients. Okay, so these are just casual slides I have added to it. After that, but it does not end here, and I have told you that if you want to detect 100% identification of the specific species of their Pseudomonas, so then you have to proceed further. And what you will do, first you will do DNA extraction, because this is the final step, and by checking the actual nucleotide sequence of the microorganism, you can easily tell that I am dealing with this such type of microorganism. So DNA extractions and its sequencing. So this is all the procedure of the DNA extraction. It is a bit lengthy process, and and it is a bit costly. Okay, it is costly, and it is a very complex procedure. But I will make videos over it, that how you will extract DNA from a cell. After that, by extracting DNA, you have to sequence it, and this is the process of sequence, and it is very complicated process. But just keep this thing in mind, that why you are performing sequencing, because you want to know about the whole DNA sequence of your microorganism. Okay, so for example, this is the actual sequence of this is the entire DNA sequence of your microorganism. So by getting this, what you will do, there are different softwares in which the hundreds and thousands and millions of sequences of different microorganisms are fed in it. Okay, are stored in it into the database, and the best database is the NCBI database. Okay, so in the NCBI database, hundreds and thousands and millions of nucleotide sequences of entire genomes of microorganism are fed, and I'm pretty sure that you will be dealing with the microorganism that is from the first that is present in that database. So you will take the sequence, you will enter the sequence in the NCBI database, and after that, it will take a 5 or 10 or about 2 to 5 minutes, and after that, your nucleotide sequence will be compared with thousands and hundreds and thousands of the sequence of microorganism. And after that, when it is 100%, it gives 100% match with the microorganism, so there will be a name of that microorganism, and there you will then you will be 100% sure that I am dealing with a specific species of a Pseudomonas. Okay, and how you will compare your nucleotide sequence in the NCBI. So I have made a video over this, and it is mostly there. Just check on my YouTube channel. I have made to 10 to 12 more videos over bioinformatics. It is related to bioinformatics. So there is a video that how you will perform blast, and it because what is blast? You have to compare and align your sequences with different other sequences. So the more the comparison between the between the nucleotide sequences of the microorganism, the more you will know that I am dealing with this microorganism. So check on the videos about the bioinformatics, and there you will know that how you will compare your entire genome with the with other genomes of the microorganism. Okay, so at the last, if we summarize our today's lecture, so what we have done, the first step is that you have to take you have to culture the sample. Okay, and after culturing the sample, okay, if I turn the color black, so if after that, first you have to sample the culture the sample. Okay, after that, when there is a growth, so then you will perform gram staining, and through gram staining you will be able to know that either my microorganism is gram positive or gram negative. Then you will check the morphology of it, that what is basically the morphology of my microorganism, either it is cocaine shape, bacillus in shape, rod shape, spiral shape. Okay, different other type of morphology. After that, you have to detect the mobility of it, that what is the mobility, either it is mobile or not. And you can also check the actual okay. After that, then you have to perform the biochemical test. So up till here, it is enough for a microbiologist when he is working in a laboratory. But if you want to proceed further, then you have to perform DNA extraction and DNA sequencing. And by performing it, you will be 100% sure that I am dealing with a specific sequence of, for example, a pseudomonas or other. And up till here, you will be able to know that I am dealing 90%. There will be a 90% chance of it. Okay. So up till now, up till here, it is also correct. But if you are proceeding further, so you can do it, but it is a bit costly, and there is a there is a bit cost of it. And if you are working in a such laboratory that that it is not that much good, so for DNA extraction and DNA sequencing, you have to send your sample to abroad or to foreign countries in order to sequence it, or to the next city. Okay, so there will be a cost over it. So this was a very brief video about the by how you will identify a microorganism in the lab, and after that, what will you do with it? Okay, so I will discuss each and every step in detail in the later videos. I have made a video over the gram staining, and after that, how you will check the mobility of it, how you will check the structure of it, and how you will perform different biochemical tests. So I will make a videos over it, then you can check it in my on my YouTube channel. Okay, so up till now, thank you very much. If you have understand this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.